Well, thanks everyone for thanks Evan for the kind introduction, and thank you to CFRI and the organizers for having us here and the opportunity to share some of our team's work. We like to say in our group that we're exploring how to apply the science of the small in terms of nanotechnologies to our tiniest patients. It's increasingly harder these days in the current climate to do big things with small science, and so hopefully we can all push the needle forward. It's just an obligatory slide for the lawyers, so they don't yell at me. But before we get started, I just wanted to highlight that I'm actually pinch hitting today. Dr. Ruby Sims, who's on the right over there, is a CFRI Elizabeth Nash Fellowship awardee in our group, who unfortunately could not make it today. So I'll be sharing a little bit about her work alongside our now Dr. Ruth Foley, who graduated about 3 weeks ago now. And she's in the Bay Area. She's just started her job in industry, and she's really been a driver for a lot of this work, and allowed our group to really enter into the CF space. So she's got big things ahead of her too. So hopefully we can share a little bit about that. So just a kind of a little big broader overview, and I apologize for the title change. I figured I'd give a little bit more of a broader talk than what Ruby was probably going to share with you. But our group really tries to focus on how do we design tools towards achieving what's called precision pediatrics. And that's really kind of a buzzword in a lot of ways, precision anything in medicine. But the way I think about it is that engineers, physicians need tools, and engineers are the folks that create those tools. And so we think about tools across 3 themes to reach that buzzword. Where in terms of precision pediatrics, we're trying to move towards a goal where we know someone's disease pathology and their state of health from the molecular level on up. In order to do that, you're going to need tools across 3 themes. The first theme in my mind is you need tools to hopefully collect a large amount of data in order to develop the precision that's implied by precision medicine. The second theme is really, once you have the data that kind of fuels this precision medicine engine, you want to have tools that will hopefully help you take that data and interpret and mine that data in new ways, and hopefully create models of disease processes that can hopefully help you feed into the drug discovery process. We think about that in terms of biofabrication types of approaches, using 3D bioprinting and other types of technologies to create different types of disease models. And the third theme is really, if you have the data, now you want tools that hopefully help you act on that data in a therapeutic sense. And we think about that in terms of thinking about solutions for manufacturing gene and cell therapeutics. And that's really where our work along along CF falls along this theme, as particular as we think about how we can hopefully make gene and cell therapies more accessible to the folks who need them. Along this kind of theme, the push towards precision medicine is really beginning to yield exciting new treatments and interventions across medicine, not just in CF or in pediatrics in general, but I'll give you a couple of examples outside the space. There's work from our colleague Don Cohn at UCLA, where we're thinking and achieving gene therapies where we're actually treating patients where we can correct disease-causing mutations for primary immunodeficiencies, such as severe ADA deficient, severe combined immunodeficiency, a disease that was previously labeled the bubble boy disease, and using some of the genetic engineering tools that have been developed over the years. Dr. Cohn has and others in the world have kind of thought about ways to address some of those pathologies using gene therapeutic approaches. As an oncologist, we increasingly think about how we can harness the immune system to attack cancers, and basically developing ways to kind of train immune cells in your body to recognize and attack different cancers. And the field's reaching the point where this is an article that was in Science about a month ago, where some of these cancer immunotherapies can be generated in vivo now to remove roadblocks to manufacturing and deployment of these interventions. And hopefully that speaks to some of the access that issues that we'll talk about in a little bit. In addition, some of you guys might have heard about some news, really exciting news from a couple of months ago, I think about in May, when researchers and colleagues at the Children's Hospital of Philadelphia reported on the successful application of a gene therapy solution for a baby with a rare urea cycle defect. This was a bespoke gene therapy that they basically went from bench to bedside in about 6 months, which is amazing. Thinking about developing the gene editing technology instructions to deliver to the cells using different types of what are called lipid nanoparticles, which we'll talk about in a little bit in terms of our work, and ultimately yielding and going through multiple in vivo model studies within that time frame in order to get to a treatment to this baby really fast. This is really an N of one kind of treatment right now, but it speaks to where hopefully science and medicine are going as we move forward. But as we think about how we can apply these types of approaches to CF, we want to make sure that we're not just treating an N of one, but hopefully moving towards technologies and solutions where we can treat an N of many. And so this is really kind of motivated the development of gene therapies to treat CF, because CF, as this audience is no stranger to, is a disease that results from a dysfunctional CFTR protein that is the result of any number of 1,700 mutations, but the potential CF disease-causing mutations. There's organ-specific morbidities that we all have to think about, particularly in the lungs. And while current treatments have revolutionized how we take care of children with CF, they're not effective for everybody with CF. And we want to hopefully close that gap. Hopefully gene therapies are one solution to provide that. As I kind of mentioned, there are different, there's over 1,700 known CF causing mutations, different ways to basically misspell the CFTR gene, and these mutations can impact a whole lot of the function of CFTR in a lot of different ways. As someone who's not an airway biologist, I'm going to kind of skip that overview, but just to say that there's a lot of potential targets for different gene therapies as we move forward. And the current treatments like modulator therapies like Trikafta have been amazing, but ultimately they are effective for, while they're effective for a majority of CF patients, about 10% of patients don't receive kind of the full benefits of these types of therapies. In particular, of that 10%, a lot of that population falls within, the non-white ethnic groups, which creates a health disparity, which we hope gene therapies can hopefully address as we kind of move forward. So how do we move towards getting to a gene therapy? What are the needs? You're going to ultimately need some kind of instruction set to correct dysfunctional mutations that cause dysfunction in CFTR. You're going to need ultimately some kind of vehicle to get those instructions to the right kind of cells, and ultimately get those instructions to hopefully some kind of airway basal stem cell or progenitor cell that's long-lived, so that that correction takes for a long time. So the approach that we're taking at UCLA is really to take a multidisciplinary approach to thinking about gene therapies for cystic fibrosis, where these are my colleagues, Bridget Compertz and Don Cohn. Their groups have specific expertise. Dr. Cohn's expertise is in developing different types of gene editing tools, and he's sort of making the cargo, if you will. Dr. Gompertz is an oncologist who has an interest in airway stem cell biology, and she sort of is the part of the team that helps us sort of identify the highest value targets cellular targets that that are hopefully ripe for correction for gene therapies. And my group is sort of the delivery group. So we're sort of Amazon, if you will. So we develop, Amazon became important to you guys because they, figured out how to put things in better boxes and to get those boxes where they want to be. The same is true for gene editing materials, right? You want to be able to package them in such a way where they get to the right types of cells to maximize their benefit. And so that's what our group kind of thinks about as we kind of move forward. And we'll kind of step you through some of our thought processes as we kind of continue the talk. So the first thing is cargo, right? You have to deliver something. And so this is really the cone group's work. But as we kind of think about it, genetic mutations, particularly those in CFTR, are basically like making a typo. If a word in a sentence is misspelled or punctuation is placed incorrectly, it can change the meaning of the sentence or end the sentence too early. And that's essentially how different CFTR mutations can work. And so gene editing technologies like CRISPR-Cas9 are sort of like autocorrect. They give you sort of the capability to hopefully rewrite or edit regions of the genome to hopefully correct those dysfunctional mutations. There are other gene editing technologies that are out there that are being considered for CFTR editing, in addition to using CRISPR-Cas9 with basically short templates that can kind of correct small pieces of CFTR. There are technologies like base editing technologies and prime editing technologies that also sort of correct regions of the gene. But for our work, we've been thinking more about a strategy that will hopefully be mutation agnostic, whereas the strategies on the other slides hopefully help you correct specific CF causing mutations. We want to think about a strategy that will hopefully give us the ability to insert an entire corrected CFTR gene cassette at the dysfunctional CFTR locus and basically overwrite it so that we can correct all types of mutations all at once. And this is really work that Don and his students, initially led by Dr. Paul Ayub, and now he's graduated and starting his own company, have been taken over by Vrishti and Colin, who are working feverishly to make the different cargoes that we're looking at. And just since this is their work, I'm just going to give you a brief summary of kind of the thought process. They're basically designing a series of guide RNAs that help the CRISPR-Cas9 system target a specific part of the genome that they want to insert the corrected CFTR template in. And ultimately, a majority of the work right now, and hopefully this is work that should be on bioRxiv in about a week or two. So I'll happy to share that when that's out. Has been involved in optimizing this CFTR donor that they've been developing for this site-specific insertion process. And so these types of donors essentially have a coding region that codes for, in our case, CFTR, and that they're flanked by different homology arms that help basically fit like Legos into the double-stranded break that the Cas9 enzyme creates when it is guided to a specific site by the guide RNAs that they're designing, and by a process called homology-directed repair, that template basically inserts into that region. And so they've been trying in their part of our collaboration to really optimize the design of this type of template. So A, it's short enough so that we can package it into our nanoparticles effectively, and B, have enough performance to yield the correction that we're looking for. And so to do this, they have a workflow where they're thinking about a process called codon optimization, which we'll kind of go into in a little bit, but kind of the idea is that they've established a set of donor templates where they can shorten those homology regions to about 50 base pairs and still achieve the similar editing performance with the templates that were that similar editing performance to similarly constructed donor templates with longer regions. Using kind of codon optimization techniques, what they ultimately see is that you get about, in their hands with electroporation, about 1 to 3% integration of that donor template when you try and insert it. I'm using CRISPR Cas9 RNP. But because of the codon optimizations processes that they've been selecting from, they're still seeing a decent amount of CFTR protein production. Ultimately, that is one that's one nice piece of data, but just because the protein is there doesn't mean it's working correctly. And so with the help of Dr. Illick, who's sitting right there, who I actually met at CFRI a couple years ago, we started a collaboration that's bared a lot of fruit, and we would not be able to understand whether or not our CFTR gene corrected cells actually work without your help. And so please. And so what they see is that with these donors, we're able to get some degree of correction of the CFTR gene, which is great for our purposes, because now we can think about how we can place them in nanoparticles. So we think we have some cargo at this point where we can do what we're hypothesizing, but also place them in nanoparticles that will hopefully be more clinically translatable as we move forward with developing hopefully a therapeutic solution for CF using these types of technologies. Just because you have the cargo, there's still a lot of barriers to a CF gene therapy. Mostly it has to do with the size of your cargo. Traditional viral vector-based strategies to deliver genes like AAV or even lentiviral vectors have packaging limits where CFTR runs up against that limit. Nanoparticles, with our nanoparticles, we have a little bit more flexibility, but it's still a consideration that we still have to think about as we kind of move forward. If you can manage to fit everything into your suitcase, you still need to get to the airway basal stem cells, which, if you take an inhaled route, you've got to get through all that mucus that Devin was talking about, and then ultimately get through the apical layer of cells to get to these cells that are kind of at the basal side. So you can either take a top-down approach, and some people are thinking about a bottom-up approach using systemic-based delivery of these types of nanoparticles. We've been kind of in both camps because for whatever reason, I'll kind of share with you a little bit at the end, if I've got time, some of our ideas on the systemic-based strategy for this. But before we get there, a little bit about nanoparticle packaging, because that's sort of our wheelhouse in a lot of ways. There are a lot of nanocarrier systems that we can think about for gene delivery. We've and others have really kind of focused a little bit more in this particular application on lipid nanoparticle-based delivery vehicles, mostly because their production is scalable. They have a modular design, which we can configure with a lot of flexibility for a lot of different clinical applications. And there's clinical precedence to all this. Everyone in this room hopefully has had a COVID vaccine. These are the technologies that have that really enabled both the Pfizer and Moderna vaccines. The way you think about lipid nanoparticle design is you've got to think about essentially the ingredients. It's like mise en place. If anyone's a cook out there, there are essentially 4 basic ingredients to a lipid nanoparticle. The first and probably most important, where a lot of the IP is in what's called the ionizable lipid, which helps basically complex the cargo, the mRNA cargo that you're trying to deliver, and helps with a process called endosomal escape, which we'll talk about in the next slide. There are helper lipids that help to reduce the potential toxicities with these types of foreign particles and they also help in the endosomal escape and particle stability. There are cholesterol type moieties or sterile type moieties that help with help to tune sort of the membrane rigidity, and also a pegylated lipid that helps prevent aggregation and extends the circulation time. So when you kind of put all that together, the art of what we're trying to do is these types of nanoparticles enter into the cell by a process called endocytosis. And as the particles are endocytosed into the cell, they are carried by this structure called an endosome. And as that endosome travels to a lysosome, which is kind of like the garbage disposal of your cells, there's essentially a pH change whereby that ionizable lipid basically kicks out the cargo and dumps it into the cytoplasm. And so the trick is to design the particles so you have that effect and release the cargo before everything kind of gets to the garbage disposal. And that's essentially what Ruth has been trying to do through the course of her PhD, is trying to hone down on a recipe for a lipid nanoparticle that will work with different types of airway basal stem cells. She does that basically by systematically changing every ingredient and using what's called a microfluidic mixer to mix all those ingredients together to create the nanoparticles. And with that approach and a lot of hard work, her best performing nanoparticle formulations with human bronchoepithelial cell line that was generously given to us by the CFF, with a small kind of GFP reporter, gets about 25% transfection using her best nanoparticle formulation. She can apply that nanoparticle formulation to other airway cell types. Those are immortalized cells. These are primary human cell types. You get a little less transfection, but still enough for us to kind of move forward with where we want to go. This kind of gave us the insight into moving forward and packaging actual gene editing reagents into the nanoparticles, and using sort of a cool trick that we borrowed from Bob Korn's lab. We used basically a stoplight approach to essentially measure, using a technique called flow cytometry, the amount of non-homologous end joining, which is sort of a default pathway for gene disruption for Cas9 versus the homology-directed repair process. So the idea is that if you have a cell that is expressing this blue fluorescent protein, if you use our nanoparticles to basically deliver CRISPR-Cas9 and components targeting that blue fluorescent protein genetic locus, if the non-homologous end joining process is favored, you basically lose fluorescence because you lose the expression of the gene. But if you insert a 3-nucleotide donor sequence using a single using a small little template, you can change that BFP to a GFP. And this is one kind of cool way that we can use to systematically tune our nanoparticles for these types of gene editing cargoes as we kind of prepare for the bigger experiments where we want to deliver larger cargoes the size of CFTR. And so using this approach, Ruth got up to about 35% HDR with a recipe, with her favorite recipe, and kind of the amount that takes into account the amount of packaging of the different materials that she's trying to develop, as she's trying to fit into the nanoparticles. And that kind of gave us confidence to look at bigger cargoes that are templates that are based off of linear double-stranded DNA, which are what we'll ultimately need to package into the nanoparticles to get to a template the size of CFTR. And so using a linear double-stranded DNA template that has encodes for a fluorescent reporter called mCitrine, she looked at designing particles to incorporate those types of cargoes. And linear double-stranded DNA has not been incorporated much into lipid nanoparticles because we tend to think about lipid nanoparticles as delivery vehicles for mRNA cargoes for a number of reasons I don't have time to kind of get into. But the idea for this experiment is she wanted to see whether or not we could package everything into the same particle and get a good editing effect, or if we needed to separate the cargoes out into two different particles. And based on her data, she actually sees the highest editing performance with particles that have the donor template combined with the Cas9 mRNA and the guide sequence. So that simplified things for us to the point where we were encouraged to take the next step and actually try to deliver a CFTR correction cassette, a donor, into the lipid nanoparticles. And again, with Dr. Illick's help, we kind of ran the same set of experiments for Western blot and Ussing chamber assays, and we see that we can get correction of CFTR using our nanoparticles. And this is up on bioRxiv right now. Hopefully it's finding a home with a particular journal soon, and hope we can kind of hopefully share that as the news comes. Sorry, I forgot to add that link. This is an old slide, I guess. What this has sort of taught us, though, and is that even with small amounts of integration, about 3 to 5% with our lipid nanoparticles, we can get up between 80 to even full correction of CFTR function using this type of approach. And so it's a combination of the lipid nanoparticles, codon optimized donors, and the right cargoes to the right cells will hopefully kind of inform how we can apply this moving forward to different systems. We also want to see how we can improve the delivery even more. I think we are hypothesizing that some of this, our lower integration is really a consequence of having nanoparticles that are a little bit more heterogeneous, that are a little bit more heterogeneic in how they are effectively packaging the different cargoes. And so using cryo-TEM, like this first speaker this morning, we're starting to interrogate that to see how we can ultimately move towards recipes where that packaging is more effective, so we can get more bang for our buck from our delivery approach. How am I doing on time? I think I got a little bit. Got it. Okay, sweet. All right. So then, in lieu of kind of going into this a little bit more, I'm just gonna, in lieu of, I'm just gonna move forward and kind of skip over a bunch of this work. But the third part is we want to be able to model these types of delivery in more realistic systems. We think the highest value targets, based off of work that Dr. Gompertz and her team has done with the CFF Stem Cell Consortium, are the target airway basal stem cells that kind of reside in the submucosal glands of the large airways. This makes them interesting targets for a systemic delivery approach because those cells have a proximity to different bronchial capillary beds, which we think we can access using the nanoparticles. And so now we're thinking about how to develop systems and that can model that delivery process as we kind of move forward. We know that without a simulated vasculature, our nanoparticles can transfect and deliver at least GFP cargoes to these types of cells within submucosal gland organoids. And now the challenge is ultimately going to be, can we add the vasculature and see how we can kind of tune how the nanoparticles can cross endothelial barriers. And this is sort of our first sort of set of experiments to do that. I don't have time to go into that. Hopefully it's a story we can share for another time, but using a combination of transendothelial electrical resistance, bulk RNA sequencing, and phosphorylation analysis, we are looking at how essentially whether or not there's changes in membrane permeability and transcriptomic profile and phosphorylation of different parts of an endothelial barrier in response to lipid nanoparticle delivery in an environment that is mimics a CF airway using a CFTR inhibitor, small molecule. And the gist of it is that we see sort of profile changes in what's called Rho GTPase cycle, as well as cell cytoskeletal dynamics, which include changes to cell cycle and mitosis, as well as endosomal trafficking in response to the lipid nanoparticles and the CFTR inhibitor kind of moving forward. And so that's basically the gist of that. And in my 1 minute left, I just wanted to kind of summarize and say that, this is sort of what we're thinking about at UCLA, is thinking about how we can take a team approach to science to come up with the right box for your gene therapy, hopefully the right cargoes for your gene therapy, and hopefully get to the right, those gene therapies to the right cells. And none of this work, I get to be the quarterback. I get to, and I should be kind of the coach on the bench today. Ruby should be up here. Hopefully she can come next year and share a little bit about the work. But these are the folks that really do all the work and kind of put up with my crazy ideas. We have to, thank the folks who pay our bills, and especially CFRI for allowing us to join your community and introduce us to good friends who help us hopefully advance our science. We're always looking for, new folks, and so shameless plug, we like to call ourselves the UCLA Nanotransformers because we hopefully transform cells using nanotechnology. Just don't tell my bosses at Mattel Children's Hospital at UCLA, because the transformers are actually Hasbro. But happy to answer any questions. Sorry if I babbled a little bit.